Welcome to the third installment of our Thrips Identification video series. In this video, we'll take a closer look at identification features and walk through how to identify thrips using a dichotomous key. In order to use our identification key, you need to use adult thrips, and you want to position them on their stomach so that their wings are facing towards you. We discussed how to tell the difference between larvae and adult thrips in part 2 of this series. Most of the identification features we use in our key are found on the head and pronotum. First, we look at the long, coarse hairs at the top and the bottom of the pronotum. These are called major setae. Most thrip species will also have smaller hairs on various parts of the pronotum. These are called minor setae. These are usually too small to see, so we don't use them in our key. However, if you are using a microscope with very high magnification, you might see some of these, and we don't want you to be thrown off by them. The next feature we look at are the ocelli. These are a trio of pigmented spots between the larger compound eyes. Depending on the species, the ocelli might be dull red, bright red, or gray. They might appear like 3 separate spots, like the example on the left, or the red pigment can blur together into a single crescent like the one on the right. A dichotomous key is a step-by-step guide that allows you to identify something by presenting you with 2 choices at a time. In our Thrips key, each step will provide you with 2 scenarios. Scenario A on the left and Scenario B on the right. Each scenario will have a detailed description of one or more features that you can use to differentiate between these 2 paths. There is a diagram above each description that illustrates one or more of the features described. At the bottom of the description, you will find where you need to go next, depending on which scenario matches your specimen. Or if you've arrived at a result, you will find a species name. Let's look at an example of how this works. Suppose we wanted to use a dichotomous key to identify what breed of dog this is. This key might start by asking what kind of ears the dog has. Scenario A says that the ears stand fully upright. In Scenario B, we see the opposing statement that the ears do not stand fully upright. This key is also giving us additional information that the ears might be fully or partially floppy to ensure that there's no confusion. The photos above each statement illustrate examples of what this might look like. We can see in this photo that the dog has floppy ears, so we would follow the directions for Scenario B and go to Step 10 to continue identifying our dog. Every step is designed to eliminate possibilities. For example, because this dog does not have upright ears, we can say that it's definitely not a German Shepherd or a Husky, or any other breed with pointy ears. It doesn't yet tell us what it is, but step by step the key eliminates more possibilities until only one possibility remains. We're about to go through an example from the Thrips key, but first let's go over some best practices that will ensure that you get the most accurate result. When you use a key, it's important that you always start at the beginning. For each choice you're presented with, make sure you read the entire description and don't only look at the pictures. Make sure you look at multiple specimens, as some features might be more visible in some specimens than others. Once you've gone through the key and arrived at an answer, read the species profile to make sure that the ID makes sense. If the options aren't making sense and you can't get a good match, you might have one of the many Thrips species that aren't in this key. When in doubt, get help from an expert. To illustrate how this works, let's identify this specimen together. We'll walk through step by step, and I'll explain some important elements of dichotomous keys as we go. The first step is asking us whether we have a pale or dark colored thrips. Option A says that the head and pronotum are light tan or yellow. The abdomen is either pale yellow, tan, or light to medium brown. Now we can immediately tell that this is true of our mystery thrips, but it's always a good idea to read the second option as well. Option B says that the head, pronotum, and abdomen are all brown to black in color. It then clarifies that the head and pronotum might be lighter brown than the abdomen, but not yellow. Okay, so we can feel pretty confident that A is the right choice. So now it's telling us to go to step 2. Step 2 is asking whether or not the specimen has wings. Let's read both options. Is it option A, very short wings, shorter than the width of the body, or option B, long fringed wings extending nearly the length of the body? This might seem like an odd question, but there are a few species that have wingless forms. If we look at our mystery thrips, we can see that it does have long clear wings covered in hairs extending down its body. This means that the answer is option B, so now it's telling us to go to step 3. In step 3, we're looking at multiple characteristics, so let's look at it one sentence at a time. In option A, the first statement is that there are no long coarse hairs on top of the pronotum. The corresponding statement in option B is that the top of the pronotum has 2 pairs of long coarse hairs that are roughly equal in length. Let's take a closer look at our specimen. Here we don't see any long hairs on top of the pronotum. This suggests that option A is the answer. But in order for us to accept this answer, all of the statements in this box must be true. So let's keep reading. The next statement is that the bottom of the pronotum may or may not have long coarse hairs. Our specimen does have long coarse hairs on the bottom of the pronotum, but this statement is telling us that even if we don't see these, that's okay too. Lastly, option A says that the ocelli may or may not be red. Again, our specimen does have red ocelli, but again, the statement is telling us that this option can still be correct even if they weren't red. You might be wondering why we bother putting in these may or may not statements. At first glance, they seem pointless. These may or may not statements accomplish 2 things. They reduce confusion, and they highlight when a feature shouldn't be used to differentiate between A and B. The statement about the hairs on the bottom of the pronotum are to reassure the reader that if you see these long coarse hairs, that those aren't the ones we're talking about in the first statement. In dichotomous keys, the statements in option A and B often correspond to point out similarities or differences between the 2 options. So we might include may or may not statements to contrast with a definitive statement that's in the other description. You will see this used most often when one of the options ends with a species. Instead of sending you to the next step, option B ends with Frankliniella occidentalis, commonly called Western flower thrips. We've added this additional detail so that you can be more sure of your answer. All Western flower thrips have red ocelli. However, not all thrips with red ocelli are Western flower thrips. So we've included this may or may not statement in option A to tell you that you cannot use the presence of red ocelli alone to distinguish between options A and B. In this step, the first sentence of these descriptions was the main feature that differentiates the 2 paths. The other 2 statements are just adding clarification. For our mystery thrips, all of the statements in option A are true, but not in option B, so we can proceed to step 4. Now in step 4, we are using the color of the ocelli to differentiate between the 2 options. Option A says that the ocelli are gray, whereas option B says the ocelli are red. Our mystery thrips does have red ocelli. The rest of the description in option B is another may or may not statement, so we can move on to step 5. The first statement in step 5, option A says there are 3 pairs of long coarse hairs on the bottom of the pronotum, and it specifies that the outer 2 are distinctively longer than those in the middle. Contrast this with option B. It says that the coarse hairs on the bottom of the pronotum are short and might be difficult to see. On our specimen, we can clearly see the hairs on the bottom of the pronotum are quite long, and the outer pairs are longer than the pair in the middle. as option A says. Next, option A says the wings are pale or absent, and in option B, the wings are either gray or black. Our specimen has pale wings, so option A is correct again. Here the key gives you some additional details about the kind of damage that they might cause in the crop, and that might help you confirm the ID. This brings us to the conclusion that our mystery thrips is most likely Thrips nigropilosus, commonly known as chrysanthemum thrips. Okay, let's look at another example. As always, we start at the beginning. We can confirm that this is also a pale colored thrips, so let's go to step 2. Step 2 is asking whether or not the specimen has wings, and when we look at this mystery thrips, we can see that actually it doesn't. We can clearly see the abdomen, and there are no wings covering it. This specimen only has tiny nubs where its wings ought to be. This is actually a very rare trait among the pest thrips we encounter in the greenhouse, so we have very quickly arrived at the answer. And yes, this is also chrysanthemum thrips. This species has both winged and wingless forms. And this is why we urge you not to skip this step. Of course, as we just saw in the previous example, if you did skip this step, the key would still bring you to the right answer, but it would take you longer. A population of chrysanthemum thrips could have both winged and wingless individuals simultaneously, so this is another reason why it's helpful to look at multiple specimens when you're IDing thrips. Once you arrive at an answer in the key, you should ask yourself if the results make sense. Remember, our simplified key doesn't include all thrips species. We've focused on the species that we're most likely to find in the greenhouse, but there are lots of other thrips species that occur naturally outside in Canada or could hitchhike on imported plant material. There are many thrips species that are not greenhouse pests, but they might still show up in your crop or on our sticky cards in low numbers. Soybean thrips, grass thrips, and predatory thrips are all common outside and often end up in our samples. If you find any odd-looking thrips, first ask yourself if you're finding a lot of them. Are they causing damage? If you can only find one or two, you probably don't need to worry about them. They might just be passing through. Other pest species have several lookalikes that require taxonomic expertise and a compound microscope to really differentiate. Many Franklinella species are difficult to tell apart. Western flower thrips cannot be easily differentiated from eastern flower thrips, common blossom thrips, or Florida flower thrips. Before you get stressed out about this, I want to reassure you that almost all of the Franklinella species we find in Canadian greenhouses are western flower thrips. If your IPM program is working, then figuring out which Franklinella species you have probably doesn't matter. But if your IPM program isn't working, and you suspect that it might be a different species, then you might want to get an expert involved. Oftentimes, though, we can do a little detective work to help us verify what species we're dealing with without needing to use a compound microscope. This is where learning more about the different thrips species comes in handy. Some clues that can help you figure this out are the host plant, geography, their behavior, and the kind of damage they're causing. For many of the species in the key, we've included some additional notes to help you do this. For our first mystery thrips, the results said this is most likely chrysanthemum thrips because although it isn't a very common pest in Ontario, it's more common than any of the lookalikes. However, you could arrive at this answer if you're looking at male Thrips parvospinus or Thrips setosus. Both setosus and parvospinus have dark-colored females and pale-colored males. So if you're seeing pale-colored females, then you can be confident that it isn't one of these two species. We explained how to tell the difference between male and female thrips in the previous video, so if you missed that one, make sure you go back and check it out. Another similar-looking species is Thrips palmi, also known as melon thrips. And here is where understanding host ranges and geography is really helpful. Chrysanthemum thrips have a wingless form, whereas its lookalikes do not. So if you've found some wingless thrips amongst your sample, you can probably stop here. Chrysanthemum thrips are native to North America, so they can come into your greenhouse from outside. Melon thrips, on the other hand, is not present outdoors in Canada, so it's likely only to be found on imports. The time of year here also matters. A local species can only invade from outside when it's warm enough for them to fly, but an imported pest has no such restrictions. We should also consider what crop it's on. Chrysanthemum thrips have a fairly limited host range. They're mainly pests on mums, asters, and gloxinia, but they've occasionally been found on strawberry, verbena, basil, and mint. Melon thrips have a very wide host range, so if you find them on a crop that's not a known host of chrysanthemum thrips, then you could be dealing with melon thrips. You'll find more information about thrips species featured in our key in our Thrips Identification Workshop workbook, which you can find on the On Floriculture blog. In the workbook, we've included some more insights into the appearance, biology, and habits of each species, along with a few reference photos. After you've used the key to ID your specimen, these species profiles should be used to confirm your ID to make sure that the results make sense. However, it's important that you don't use the profiles or the photos that we've included as the sole identification tool. You should never just look at photos to ID thrips, because they're a very unreliable means of identification. Thrips can vary in appearance a lot, as these photos demonstrate. Western flower thrips and onion thrips have a wide variety of color morphs that can change in response to environmental conditions. The lighting that the photo was taken on can also alter how the colors appear. Humans are a very visual species, so there's a tendency for beginners to be distracted by other things that they pick up on in a photo that might lead them astray. For example, your eye might be drawn to the striped appearance of the abdomens of the thrips in the top half of this image. However, these pale bands are not identification features. They're actually the connective tissue between the abdominal segments. These specimens had been stored in liquid before the photos were taken, and their bodies have swelled, causing their abdomens to stretch out. This can make them look quite different than they do when they're alive, or if your sample is very fresh and hasn't had time to swell up yet. We have chosen features for our key that don't change and can reliably lead you to the right answer. In some cases, we've included multiple paths to lead you to the same species to account for these variations, as we saw in our example with chrysanthemum thrips. For species with both light and dark forms, we've included them both in the pale and dark species sections of the key. Thrips species ID can feel overwhelming for beginners, but rest assured that in most crops, the overwhelming majority of thrips will be western flower thrips or onion thrips. Most of the unusual species we see are not native to North America, so you'll want to be most vigilant with imported plant material. But in established crops, if your IPM program is working well, you probably don't need to worry about it. However, if you are experiencing an unusual outbreak, particularly if it's in a crop that doesn't usually have thrips problems, then you should definitely take a closer look at your species. You'll find more videos that will help you sample and identify thrips on the Greenhouse IPM YouTube channel. Our Thrips ID key and other workshop materials can be downloaded from the On Floriculture blog. Thanks for watching. The Thrips Identification Workshop and video series was created in collaboration with Vineland Research and Innovation Centre, the Ontario Ministry of Agriculture, Food and Agribusiness, and Agriculture and Agrifood Canada.